right, class, this is our sheep eye dissection, and here's our sheep eye. And we can see the anterior structures there. We flip it onto the back. Here's our posterior structures, lots of tissue back here depending on how they cut the specimen. So all the yellowish areas that we see, kind of yellowish whitish areas, that's fat tissue or adipose tissue. And then we have the kind of brownish structures there. That's going to be our muscle tissue. So we can see the muscles attaching to the eye. Sheep eyes, they have 4 extrinsic muscles, skeletal muscles, and in humans we have 6. So if you remember from your packet, we have 6. Now I'm just going to start kind of trimming away a lot of this tissue back here posteriorly. I really don't need to worry too much about cutting into the actual interior of the eye because the sclera is incredibly tough. The sclera is the white part of our eye that we see when we look at each other. It also makes up the outermost layer or tunic of our eye in the sheep eye. Lots of similarities between the human eye and the sheep eye. What I'm kind of looking for as I trim back here is the optic nerve, where all the information that's gathered inside the eye leaves the eyeball and heads to the brain. Sometimes it's hard to find, like in this one right here, because there's so much tissue back here. There it is. There's our optic nerve right there. And again, that's where all the information from the rods and cones is going to leave the eye and head up to the brain. Particularly, it'll hit the thalamus and then down to the occipital lobe. Okay, anteriorly, we have the cornea. Okay, very clear. Well, it's not so much clear in the preserved specimen because it's preserved. A lot of times the preservative fluid causes the once clear cornea to cloud up a little bit. Also lack of oxygen. The organism is no longer living, and so lack of oxygen causes that cornea to cloud up. We know that in living specimens such as us or sheep, it's very clear, very transparent. You can see through it very easily. It's also very curved, right? And that's going to be a part of the key to getting the light refracted onto the lens and then refracted again onto the back of the retina. Okay, this again is the sclera, the white part of the eye that we see when we look at each other, and that follows all the way to the posterior side. Okay, to cut into this, we're going to try to separate anterior from posterior eye. So we are going to cut this way. Some fluid will probably come out. I don't know if you can see that fluid coming out. That's probably going to be the aqueous humor. It's dripping down. I don't know if you can see it, but anyway, that's the aqueous humor, and that's going to help to give the eye structure. It's going to help to keep the shape of the eye a nice rounded sphere. One thing also you'll notice about the sheep eye is its cornea is very kind of oval, where ours is very circular. So that is another difference between the human and the sheep. Well, I didn't line that up very well. There we go. Try to remove some of the tissue. All right, so when we open it up, okay, there we go. So again, here's our anterior chamber. Here's kind of the middle and also the posterior chamber. So what we see here, this is the lens. Okay, it's very opaque, very hard in the preserved specimen. In the living specimen, even in sheep, it's going to be kind of not so hard. It's not super squishy, but it's kind of somewhere in between. There is a little bit of give to it because as the ciliary muscles or the ciliary bodies, those are muscles, it's smooth muscle, it's going to pull and kind of twist and turn that lens to again refract light just so that it can hit the retina the way it's supposed to based on how close or far away the image is. Let's see if I can get this to completely separate. Looking again at the anterior, here we can see those ciliary bodies. Where's my probe? They look almost like mushroom gills. So if you look in here, all of these little mushroom gill little things, going all the way around, those are ciliary bodies. Okay, in our diagram, it looks like they're just one on each side, but really it's all the way around. And then you can kind of see the at this point, translucent cornea there in the middle. There's some fluid in there too. But that aqueous humor, that's going to lie between the cornea and the lens. Okay, it's a fluid. That's all the fluid that is now on my paper towel. And that's going to help keep the shape of the eye for the sheepin and for us a circle. Okay. Going back to the ciliary bodies, these smooth muscles here, again, they're going to attach to the lens via some suspensory ligaments, which we can kind of see around the lens here. So these black little ligaments here, they almost look like they're kind of coming out a little bit, almost like rays of sun outside of the lens. Those are suspensory ligaments, and that's what's going to hold the lens to the ciliary body muscles. Okay, now all of this kind of fluid stuff, it looks like jelly-like substance, it's squishy like a jelly-like substance. That's all the vitreous humor. Okay, so that's the posterior chamber humor. It's not quite as fluid or watery as you can see the aqueous solution was, or the aqueous humor, more gel-like. Okay, so now if we try to remove the vitreous humor, again, there's the back of it, very jelly-like, helps to keep the shape of the eye. Okay, now we have the posterior eye. Now there's three structures to it. First of all, we have the sclera, all the white stuff, all the way around. If we could remove all the fat and all the muscles, we would see the sclera. You can kind of see it here, but it's the white part of the eye. Deep to that, we have the choroid, which is a black structure. You can see a pretty good chunk of it under here. And the choroid is black, it has lots of pigment in there. That is going to keep the light from bouncing around the eye once it passes the lens. Okay, we don't want the light bouncing all over the place. That's going to cause some issues. So choroid, so sclera on the outside, choroid, and then finally the retina. This very delicate membrane, it's even a little bit see-through, that is the retina. Okay, now if I disturb it a little bit, well, a lot, and try to gather it, we'll see that it sticks in one spot. So as you move it around, where it won't move, okay, you can kind of see it moving around that one spot there, that is the optic disc. Okay, the optic disc, another name for that is our blind spot. It's basically where the retina attaches to the back of the eye, okay, to the choroid, to the sclera. And all the information concerning vision leaves the eye through this optic disc, and that should be very consistent with the optic nerve here in the back that we had seen earlier. So if you can see that, that is the optic nerve, and that's where the information goes to the brain. Okay, so there are no rods or cones, no photoreceptors at all, which we know the retina is full of photoreceptors, rods and cones. Well, at this one spot, optic disc blind spot, there are no photoreceptors. So that's why it's a blind spot, because there aren't any of those receptors picking up any kind of light or images. Okay, if we remove the retina, we can get that out. Then we can just see the choroid, that middle layer. In the choroid, as you can see, it has a very shimmery, kind of iridescent to it. We don't have that. Okay, this very iridescent, kind of shimmery, kind of cool, real pretty looking. And a lot of these sheep, it's more kind of like a greenish blue, real pretty. This one's more yellow. Anyway, that's what's going to give them night vision. Remember how we talked about like humans can see a little bit in dim light or mostly dark light, but we really can't see well in dark light or darkness because we're relying on our rods. And we don't have this thing here called the tapetum lucidum that animals have that are nocturnal. They can see at night. So if you've ever seen shone a light or shined a light into your dog or cat, or if there's a deer on the road and deer in headlights, it stops in your headlights, it almost reflects back like a yellow or a green or an orange. It's different depending on the species of animal because their tapetum lucidum is slightly different in each species. So our choroid layer, because that tapetum lucidum is embedded in the choroid layer, our entire choroid is just black. Okay, lots of pigment there, no tapetum lucidum. Okay, so if we go back to the anterior part, I'm not sure. Sometimes you can see the iris and sometimes you can't. It just kind of depends. But if I try to cut through here, and again, the iris is what gives us our color of eyes that we have. Most sheep have blue. Most sheep have brown eyes. And there you can see a little bit of the brown, and that is their eye color, that is the iris. Now anteriorly on the iris, there are 2 layers of smooth muscle. Okay, autonomic nervous system controlled muscle. And that's going to allow the pupil to dilate or constrict depending on how much light is coming through. Okay, so 2 layers of muscle on the anterior side, and then we have the ciliary bodies, these muscles back here that attach to the lens.